Sensory receptor physiology. The relationship between stimulus strength, graded potentials, and action potential. Stimulus strength refers to the intensity or magnitude of a signal that excites a neuron. Graded potentials are changes in the memory potential that occur in response to a stimulus, and they can vary based on the amplitude, duration, depending on the strength of that stimulus. Graded potentials are considered local signals that occur in the dendrites and cell bodies. When a graded potential gets strong enough, it's going to be able to reach the threshold to depolarize, and then an action potential can be stimulated. Action potential is this rapid but brief electrical signal that travels down the axon of a neuron, eventually leading to the release of neurotransmitters down at the axon terminal. So in summary, the strength of a stimulus can lead to graded potentials that may trigger an action potential if the stimulus is strong enough. At the bottom of the axon hillock in the first node arambier, you'll see that unless the graded potential meets that dotted line, it will not fire. Once it meets the dotted line, it becomes these spikes that represent action potentials. And as it travels down the cell membrane, eventually causes a release of neurotransmitter. Analog to digital conversion, also known as ADC, is a process of converting this continuous analog signal, such as a stimulus or the graded potential, into a digital signal, which is going to be the frequency or number of action potentials. As you can see over here, there's a lot more action potentials in this picture than in this picture on the left. So the strength of a stimulus or graded potential isn't translated into that frequency of action potentials. This means that a neuron receives a weak stimulus or weak graded potential, it will generate a lower frequency action potential. Whereas a stronger stimulus or stronger graded potential, it will lead to a higher frequency of action potentials. Recruitment and range fractionation. Recruitment and range fractionation are two mechanisms by which the nervous system can encode the strength of a stimulus using the firing rates of multiple neurons. Recruitment refers to the process by which increasing levels of stimulus intensity activate additional neurons that will trigger more action potentials. This means that a weak stimulus may only activate a few neurons, whereas a strong stimulus will activate many neurons. By recruiting more neurons, the nervous system can increase the overall firing rate of neurons and encode a stronger stimulus. On the right-hand side, range fractionation refers to the process by which the range of stimulus intensity is encoded by different types of neurons. So you have a neuron A, B, C, and D, and they have different ranges. This means that low-range stimulus intensities may activate a specific group of neurons that has a low threshold firing, whereas a higher range of intensities may activate a different group of neurons for firing with a higher threshold. The letter A represents a lower intensity neuron, and letter D represents a higher intensity neuron. For example, inside the eyes, we have the retina, and there are two photoreceptors that contribute to range fractionation. There's the rods and there's the cones. The rods are a little bit higher sensitive to light, whereas cones are less sensitive to light, but they can detect color vision. Audition inside the cochlea of the inner ear also has different types of hair cells, and they can respond to different frequencies of sound. High frequency sounds are located towards the base of the cochlea, whereas low frequency sounds are detected at the apex of the cochlea. Lateral inhibition is a neural mechanism that enhances sensitivity and resolution of sensory cells by sharpening the contrast between adjacent stimuli. This occurs when the sensory cells are arranged in a linear format, such as inside the skin or the retina of the eye. The process of lateral inhibition involves inhibiting inner neurons that are connected to adjacent cells. So when a sensory cell is stimulated, it sends an excitatory signal to both neighboring cells, and to inhibitory neurons. The inhibitory neurons then send signals that are inhibitory back to the adjacent cells to then reduce the response to the stimulus. Therefore, this inhibition creates a sharpening of the contrast between the activated cell and the neighbor cell, allowing more of a precise discrimination of that stimulus. So right here you can see when a pin is inserted into the skin, and neuron number B sends a signal, and these neurotransmitters are going to be released, but the neuron B will inhibit neuron A and C, heightening the signal of neuron B. So on the right-hand side, you'll see that action potential frequency in the neuron also changes. There's a lot more frequency over here in the middle, and neuron B than there are in A and C. And you can see the graph where it's a lot higher in letter B. As a result of inhibition, it will then depress neuron A and C by even lower amounts, making neuron B a lot more precise and able to discriminate in the location of signal. Somatosensory pathways. Somatosensory pathway is a dorsal column pathway. This is a somatosensory pathway that transmits information about touch, proprioception, which is sense of body position, and vibration from the body to the brain. The pathway consists of three main areas: the sensory receptors in the periphery. They then relay signals to the nuclei inside the spinal cord, and then the thalamus and the primary somatosensory cortex to the brain. Sensory neurons are the cell body in the dorsal root ganglia, and they project toward the dorsal column. They will then synapse with interneurons located inside the medulla, the brain stem. These are the first order neurons. The inner neurons are axons that ascend the brainstem through a structure called the medial lemniscus, and they synapse to the third order neurons inside the thalamus. So you can see right here that there's a crossing over called decussation, and that goes through the brainstem and it crosses over to the secondary neurons shown in green, all the way up to the third order neurons inside the thalamus. And these third order neurons will then project directly to the somatosensory cortex for perception. So overall, this is a highly organized, specialized somatosensory pathway that allows for precise detection for discrimination of touch, vibration, and proprioception. The anterolateral pathway, also known as the spinal thalamic pathway, is a somatosensory pathway that transmits information about pain, temperature, and crude touch. The pathway consists of three main stages. Again, the sensory receptors in the periphery, relay nuclei in the spinal cord, and then the thalamus to the somatosensory cortex. So it begins in the dorsal root ganglion, and they synapse to the interneurons of the dorsal horn. So you can see right here, they come in, and then they synapse to the dorsal root horn. The interneurons have cell body where they'll decuss from spinal cord, so over here it crosses over, goes up all the way to spinal cord on the same side, and then projects into the thalamus, eventually to the third order neurons into the somatosensory cortex for perception. As we age, our sensation also decreases. Declining sensitivity of the sensory structures, such as vibration, touch, proprioception occurs. There's also an increased threshold for action potentials to be generated, so we're less sensitive. There's also more stimulation required to detect a particular sensation. There's also a decreased chemoreceptor sensitivity and also decreased response to hypoxia. In general, there's going to be a lower sensitivity to pain versus when you were younger.